The identification of therapeutic options for rare diseases represents a major challenge and a moral duty for the scientific community. Approximately 7,000 rare diseases have been identified, and collectively they affect more than 350 million people worldwide. A therapeutic option is available for only 5% of rare diseases, and due to the extremely small individual markets, orphan diseases are of limited interest to the biopharmaceutical industry. Even if genetic diseases are so heterogeneous, the molecular causes can be grouped into a handful of classes, and in most cases a gene dosage alteration takes place. For this reason, rare diseases that share a common molecular etiology could benefit from the same therapeutic strategy. For instance, the identification of drugs restoring physiological protein levels represents a promising therapeutic approach for all genetic disorders associated with genomic duplications. In our case, ADLD has been exploited to design, set up and validate a therapeutic option for genetic diseases due to gene dosage gains. Autosomal dominant adult onset leucodistrofia is a fatal genetic disorder associated with a symmetrical demyelination of the central nervous system. ADLD is caused by an excessive lamina B1 production due to gene duplications. Indeed, ADLD patients carry 3 lamina B1 alleles in their genome and not the physiological 2 copies of the gene. For ADLD, no treatment is available, and all medical interventions are palliative. In recent years, the use of synthetic small interfering RNAs has become a popular choice for the development of gene silencing-based therapies. However, a major drawback is dysregulation and excessive transcript reduction of the target, which may lead to pathogenic consequences. This concern is because dosage-sensitive genes often cause pathological phenotypes either in excess or in defect. Allele-specific RNA interference is a variant strategy that allows to downregulate a single mutant allele with minimal or no suppression of the corresponding wild-type allele. We considered allele-specific RNA interference as the best gene silencing strategy to target genetic diseases due to gene duplications, because it will avoid an excessive downregulation of the target gene. Here, a schematic representation of the rationale of our therapeutic strategy for ADLD. We identified a frequent polymorphism located in the lamina B1 coding sequence. The 85% of ADLD patients tested in our survey are heterozygous for that SNP. We demonstrated that in ADLD patients, the 3 lamina B1 alleles are equally expressed, and therefore we decided to target the non-duplicated allele of the lamina B1 gene by allele-specific siRNAs, with the aim of reducing expression close to a wild type. In the best scenario, following allele-specific siRNA treatment, only 2 alleles of the gene will be transcriptionally active, as in control. We identified 4 siRNAs able to selectively silence the target allele with minimal or no suppression of the corresponding wild-type allele. Those siRNAs have been validated on patients-derived fibroblasts. Allele-specific siRNAs restore physiological lamina B1 messenger RNA levels, silencing specifically the target allele. Allele-specific siRNAs restore physiological lamina B1 protein levels, and they ameliorate ADLD-specific cellular phenotypes such as nuclear anomalies and transcriptome alterations. Finally, allele-specific siRNAs have been tested on a disease-relevant patient-derived cellular model, namely directly reprogrammed ADLD neurons. The siRNA treatment restores both lamina B1 messenger RNA and protein levels close to control levels and abrogates the ADLD-specific phenotypes. In conclusion, our patent represents the first therapeutic option for ADLD patients and represents the first therapeutic strategy for a genetic disease due to gene or genes duplication. This approach can be exploited to treat patients with a Mendelian or a syndromic genetic disease associated with a genomic duplication and having a coding heterozygous polymorphism in the target gene.